Hello, everyone, and welcome to our latest Tech Talk, Handling Unexpected Biocompatibility Test Results in Medical Device Development. I'm Amanda Hosey, your moderator for this program, and joining me is Audrey Turley, the Director of Expert Advisory Services at Nelson Labs. Audrey has more than 25 years of experience working in research, laboratory, and test design functions in the medical device industry. She is a biocompatibility expert and an active committee member of several working groups within the Association for the Advancement of Medical Instrumentation and the International Standards Organization. Audrey was responsible for the in vitro mammalian chromosome aberration assay development at Nelson Labs and was on the team that developed and validated the in vitro irritation assay for medical devices. Questions for Audrey may be submitted using the Ask a Question box on screen, and they will be supplied to her for follow-up later. Also note that slides may be downloaded under the Event Resources tab. Now, onto the presentation. Welcome, Audrey.
Thank you, Amanda. Happy to be with everyone today and talking about the unexpected that can happen with biocompatibility tests. So my goal today really is to shed some light on this situation. So when we think about biocompatibility testing, that's happening at the end of a device development phase. So this is after all the materials have been selected, the device design has been locked, we have our packaging, we know how we're going to ship everything and how it will be sterilized. So this is the end of the road test, and even with careful planning, we can sometimes see failures occur. So, we can't always predict everything, how it will play together, all the different materials and processing steps that we use. And sometimes it is best to include some of these compounds that would produce a failure, but also produce benefits to the patient. So I just want to say, when you see a failure in biocompatibility, do not despair. It does not mean you need to go back and revamp your device necessarily. I just spent some time with customers who have, seen a failure and then spent months investigating it, when really it was okay that there was a failure. It was telling us something about the device, and we could speak to, is that a risk to the patient? So, talking about investigations and how to go through these failures is really important and helpful. So the first thing we want to think about is to get into the biocompatibility mindset. Our ultimate goal for everyone in the industry, whether you are a laboratory, a regulatory agency, or a medical device manufacturer, is we want to protect patient safety. This is where all of our interests can align. So when we think about these failures, or when I dive into these, that is my first goal, is I'm always going to keep that at the top of the list and tie everything back to patient safety. So things that really help as we ask these questions is knowing specific use of the device. So how many devices could be used at one time? Sometimes we think, oh, well, we're just marketing, some things can only be used in limbs. Okay, we have 4 limbs, so we know 4 is the worst case use, but some of these are marking maybe tissues internally, so we could have up to 10. We just need that kind of information to help us understand a worst case scenario for these addressing failures. Do we have repeated use? So is the device used for 14 days and replaced and continue to be used? So we want to make sure we understand the actual clinical use and duration of use. Is it used for 30 seconds, but we're under that limited category, so it's less than 24 hours? Knowing the difference between 30 seconds and 24 hours is really important when we talk about failures in testing. And testing details matter. We want to tie those test results and how the device was tested back to patient exposure, because again, the ultimate goal is to speak to patient safety. So with that in mind, we want to dive into some of the questions we want to ask for investigating our failures. So we often say curiosity kills the cat, but curiosity finds the culprit. So curiosity is rewarded in science, at least that's how I look at it. So I love to ask a lot of questions, because it will guide me to what the actual cause of that failure is. So the first thing we want to think about is, was the testing necessary or appropriate? And really, what I'm asking is, what question are you trying to answer, or what are we trying to solve? So I had a situation where a farmer came to me, a rancher, and he had a tool that he would use with his cows to track their digestion. And he wanted to test the device after it had been used by an animal. And he tested it in cytotoxicity. Of course, it failed. And he said, "Why would it fail?" And I said, "Well, we're not really testing or getting the answer that you're looking for. And because everything in this animal would have caused several interactions with the test system that would have caused it to fail. So this wasn't really setting up a successful test. So make sure we understand that the data you get is going to actually answer the question you're trying to establish. If it's a final finished device and you're just doing the testing to get through regulatory, that's easy because then we can say, okay, that testing was needed. Let's jump to our next question. The next one I want to talk about is, was the failure unexpected? So oftentimes we'll have devices where, this is very similar to something already on the market, so I know it's safe, but it still fails a test. But oftentimes the original device would have failed too. So we want to look at what those materials are. If you have anything that's like an antimicrobial agent on your device, it's absolutely going to fail a cytotoxicity. That's just one of our biocompatibility tests. But antimicrobial agents aren't selective of the cells that they kill. So they're meant to kill bacteria. They will also kill the cells here. So we want to still do the test, show the failure, and then maybe tie it back to that antimicrobial agent. So we would do some different investigative testing. And in the end, again, tie it back to patient safety. So, and if you weren't expecting the failure, have a call at the lab and say, hey, here's the materials on our device. Do you expect any of these to have caused that response? And we have a list of known culprits for several of our tests. Okay, continuing on this series of asking questions. What portion of the device was tested? Meaning, did we test non-patient contacting portions that may have electrical components that would cause adverse responses, maybe some sensitization responses, but won't have contact to the patient. So if it failed the test, that result really isn't speaking to patient safety. Also, was the device cut? Should we have cut it? If you don't tell us to not cut, if we need to cut a device to fit into a container, we're going to cut it in the laboratory, just assuming that, hey, this is what we need to test. And sometimes we can expose wires like zinc or copper, and those things will definitely elicit some adverse responses in our tests. And was there any prep we should have included in the test that will be done in the clinical setting? Like, will the device be rinsed? We would want to make sure we rinse that also before we test it. Next set of questions to ask is, how was the device extracted or tested? Did we immerse the device? Should we have flushed the device instead, because only the internal components of the device will have patient contact? Was the extraction time too long? If it failed after extracting for 72 hours, could we cut that extraction back, especially if the patient exposure is, 30 seconds, but we're extracting for 72 hours? Maybe we could extract at 24 hours, still showing an exaggerated extraction. Was the temperature too high for the material of the device? Again, understanding those materials is really critical and important for a med device manufacturer to then convey to the laboratory, because we aren't experts in your device, we're experts in the testing, so we want to bring our expertise together. And the next is, was the application clinically relevant? Is this a topical device that was extracted and then injected into an animal? Could we do more of a surface analysis? Again, asking more of those details about the testing versus how the device will be used in a clinical setting. After we've asked all these questions, one thing I just want to highlight is we want to document what we find in these failures. Sometimes we have a tendency to sweep this under the rug, and we don't want to do that. So tell your story, tell what you learned, do good investigation, and then also connect to the clinical use and cover expectations of the regulatory agencies. And after all of this, if you find that you need help, phone a friend, or email is probably easier, but I've put our advisory services email here if you want to contact any of our experts as you deal with any type of biocompatibility failure we're happy to talk through how to address that and do your investigation. Back to you, Amanda.
All right, thank you so much, Audrey, for that interesting presentation. And as a reminder to the audience, you can download these slides under the Event Resources tab on screen. In just a moment, a survey will pop up. Please tell us what you thought of today's program by answering a few quick questions. This on-demand recording will be available for the next year. Once again, we thank you for watching.